A major new longevity breakthrough just dropped. Scientists took skin tissue from a 74-year-old donor, treated it overnight with a specially engineered repair protein, and pushed one key marker of age-related damage below the level found in skin from a 31-year-old donor. Yes, that result is real, and this may be much bigger than wrinkles or skin care. The same approach also removed large amounts of damage from old artery and eye tissue, pointing toward a new kind of longevity medicine that does not just slow aging, but repairs damage already built up over decades. As we age, some proteins in our bodies slowly become damaged. Proteins are biological tools and building materials that help form our skin, blood vessels, muscles, and organs. Some are replaced quickly. Others, especially collagen in skin and blood vessels, can remain for many years and collect damage over time. One common form appears when sugar molecules slowly attach to proteins, similar to food browning during cooking, but happening inside the body over decades. Scientists call the result an advanced glycation end product. We will simply call it sugar damage. The specific type targeted in this study is called CML. Think of it as chemical grime attached to important proteins. As it builds up, proteins may become stiffer, work less effectively, and promote inflammation. Until now, scientists believed the body had almost no way to remove this damage once it was attached. The researchers wanted a biological cleaning tool that could remove CML without destroying the protein underneath. That tool is an enzyme, a protein that performs one specific job. The team called its new enzyme CM-lase, but the perfect version did not exist in nature, so they had to build it. Computers searched through tens of thousands of known protein shapes for one that might reach the damaged part of a protein. Think of it as searching through thousands of keys for one that almost fits a lock. The researchers found a promising starting point, then created hundreds of millions of slightly different versions and kept the best performers. After 5 rounds, they produced CM-lase, an enzyme capable of removing much of the sugar damage while leaving the protein underneath largely intact. The researchers then tested the enzyme on preserved human tissue. In eye proteins from a 64-year-old donor, the measured damage fell significantly. In artery tissue from a 75-year-old donor, it fell by more than 70%, and then came the result behind the viral headline. In skin tissue from a 74-year-old donor, the enzyme removed more than half of the measured CML damage. After treatment, the remaining level was lower than in untreated skin from a 31-year-old donor. The skin result makes the best headline, but the artery result could eventually matter more. As we age, long-lasting proteins in blood vessel walls can become damaged and stiff. Older arteries lose flexibility, increasing pressure on the heart and contributing to cardiovascular disease. If scientists can eventually remove this accumulated damage inside living arteries, they may be able to restore some of that lost flexibility. That has not been demonstrated yet, but it shows why this research reaches far beyond cosmetics. The same approach might eventually be explored in the eyes, skin, blood vessels, joints, and other tissues built from long-lasting proteins. There are major limitations. The researchers used very thin pieces of preserved tissue, not living people. Because the samples were so thin, the enzyme could easily reach the damaged proteins. Reaching the same targets inside living skin or deep inside an artery would be much harder. The enzyme is based on a bacterial protein, so the immune system may recognize it as foreign. Scientists must find a safe way to deliver it and prove that it does not harm healthy tissue. And CML is only one type of damage that builds up with age. The study did not show younger-looking skin, fewer wrinkles, restored artery function, or whole-body rejuvenation. Aging involves many problems at once. This experiment repaired only one of them. Most treatments try to slow the creation of new damage, like preventing more rust from forming. This study explored something different, removing damage that had already accumulated over decades. That is the real breakthrough. It suggests that at least one form of age-related molecular damage may not be permanent after all. Scientists have moved from asking whether this damage can be removed to asking whether it can be removed safely inside a living human body. The 74 to 31 comparison is not proof of age reversal, but it may be an early sign that future longevity medicine could do more than slow aging. It could also repair part of the damage aging has already caused. If this technology reaches human trials, which target should come first? Stiff arteries, damaged eyes, or aging skin? And should health benefits take priority over cosmetic ones?